Hello everyone, welcome. This is Mr. Vona's second time recording this video. The first time I recorded it, I had my video camera right up there, bolted to the table that I'm writing on, and every time my hands moved, and apparently watching it, I talk a lot with my hands, I would shake the camera. And on the advice of Mr. Costa, who I trust, whose opinion I trust, he said, no, you have to do it again. You can't get away with that, even though it's just the first one. So this is round two. So the topic of today's video I have on this sheet of paper over here. First thing I want to say though is, guys, it is the first one. My goal is to get better if we're going to do any more, if we find that this is useful, that this helps clarify. So you have to let me know whether it is worth it or not. My goal is, the last one also was very long, it was about 35 minutes. My goal is between 15 and 25. I don't know how it's going to end up. This lesson usually takes an hour and a half in class, so I'm going to try and tighten it a bit. But let me know if it's worth it, because if it's not, then we may find another way to do this, or I may just make really short snippets for people as needed. So here's my list, and I chose this. I chose writing things down because I love writing on the board. I do have tons of good stuff for you already posted online. I have all the handouts. I have videos ready for you. This is more for the Mr. Vona style of doing what we did in this week's work. And what we did in this week's work was we looked at nucleotides, nucleotide structure, and we also looked at the structure of the double helix. And this follows with what you did in the movie. So keep in mind, this is the first one. And if you didn't watch the movie, if you didn't do the assignment, if that's something that you skipped over thinking that it wouldn't be worth your time, or you didn't take my warning about it gets good in the second half, you didn't take that seriously and just decided not to watch it, I think you should go back and watch it. It is absolutely worth it. I enjoy it. I watch it every year. I have a grade 12 class. I'm going to watch it again for you. I watch it for fun. It is absolutely worth it. It's a bit of a struggle the first 20 to 25 minutes just with names and with accents and with places, but you should watch it because it gives you a lot of good insight for the lesson that we're going to do right now. Number 3, you should have ready with you your course notes, the booklet of course notes, or the handouts that are on week 3 on D2L. Those are things that I'm going to be referring to. I have some sheets here at the side that I'm going to put in front that just show things much better than I can on this pad of paper here. So have that ready to go. If you need to pause the video now and go get it, then do that. Number 4, my fine level details may not match the fine level details you see in the textbook and may not match the fine level details you see in the videos or even on some of the sheets that I have at the side. And that's okay. Unless it's a big mistake, you can trust that they are variations of the same thing, variations of the same chemical, maybe a slightly different word that I learned or that your textbook uses or that the videos use, but it is most likely the same thing. If you notice something glaring, if you notice a big thing, sir, you said this and we see this somewhere else, please let me know, because if I'm wrong, that's the way that I'm going to find out and I'll fix it. I'll post something to give you guys an update. Number 5, DNA names. Okay, so in the last sheet, by the way, just so you can see, I wasn't joking. I actually did this lesson already, 35 minutes, and I started with this up here. So these are the names of various objects related to DNA that you have probably heard in another class. You've almost definitely come across chromatid, chromatin. You've definitely come across these last year. You've definitely come across them in possibly some of the videos that you watched already here. Allele and gene. Okay, so take a minute or two. Pause the video, look at these words, and see if you can come up with a definition for each of them. Pause now, really try it. If you had a hard time, I don't blame you. These words are complex and sometimes confusing, especially when they're used without the right context. And when you guys move from grade 11 to grade 12, we start to focus in much more on this object here. We focus much more on DNA. And that's what all of these other things are. These other words are just different ways of talking about DNA in different contexts, in different situations, in different forms, in different shapes, for different purposes. And we start there. We start here in this unit. We're going to build off of this. We're going to get much more complicated than what you guys did in grade 11. These words are important. There's nothing wrong with knowing them. We're going to use gene a little bit. We're going to use chromosome a little bit. Allele, not so much. Chromatin and chromatid, probably not at all. They are important words, but again, everything is just from DNA. So when we talk about DNA, we're talking about a molecule called deoxyribonucleic acid. This molecule is a really important molecule to life, but it actually doesn't do anything on its own. DNA is very stable. That's one of the things that makes it very useful for us. It's a very stable molecule. We can pull molecules of DNA out of organisms that have been dead for thousands of years, possibly even hundreds of thousands of years. Take that DNA out, read it and replicate it and do things with it. Very, very stable. The fact that it's stable means, or one of the side effects of that, is that it doesn't really do anything. There's another molecule that's very closely related to DNA, and that molecule is called RNA. RNA is, in my opinion, a much more interesting molecule than DNA in the things that it does. Not as important. DNA is very, very, very important, but RNA actually does some stuff, and it can fold into shapes that are a little different than the shapes that DNA can fold into. And RNA is ribonucleic acid. Okay, and you can see here from their names where all of this comes from. DNA is deoxynucleic acid, and RNA is ribonucleic acid. And so you have DNA and RNA. So we're going to draw the structure to see how we can figure out, or how the names came to be in the first place. So DNA, deoxyribonucleic acid, has a phosphate group. And the phosphate group is bound to a 5-carbon sugar molecule. Notice that there are 5 carbons, 1, 2, 3, 4, 5, and they're numbered starting from this one over here, and we'll see why in a second. 1, 2, 3, 4, 5. And the oxygen that makes up part of this ring is not counted as one of the 5. Obviously, it's an oxygen. And carbon 1, we label it as carbon 1 because it's connected to a molecule called a nitrogenous base. And I have over here the 5 nitrogenous bases. This is one of the handouts in your textbook, in your course notes rather. And one of the things that I think I included in the week 3 tasks. So we have 1, 2, these molecules up here are called purines, adenine and guanine. And then at the bottom we have cytosine, uracil, and thymine, and these are pyrimidines. Now the way that I keep track of which one is which, there are a few different ways, but the purines, or the naming system, which ones are purines and which ones are pyrimidines, purine is the larger molecule, but it has the smaller name. This is a double ring and a double ring. And pyrimidines are the larger name, but they have the smaller molecule, so a single ring instead of a double. Notice over here that I've highlighted DNA only for thymine and RNA only for uracil, so you can see that on your handout probably a little more clear than you can see it on mine. So DNA uses these 4 red, what we call bases, or nitrogenous bases. RNA, the structure of RNA is different in and of itself, but also instead of having thymine, RNA uses uracil. So these two are changed between DNA and RNA. This is RNA, uracil, and DNA uses thymine instead. And they're very similar molecules. You can see the differences right here on carbon 5. This one has a hydrogen, and thymine has a methyl group. Okay, so these are our bases. So I'm just going to draw it as a rectangle right there. Keep in mind that we're referring to these shapes here. All right. Now, there are more pieces to this molecule that I didn't draw. There are some negative charges to the phosphate. These carbons have hydrogens connected to them. Carbon number 3 has a hydroxyl group, an OH, and also a hydrogen in there. There's a hydrogen over here, 2 hydrogens on carbon 5. All right, and so this would be the complete structure. I usually don't draw those hydrogens because they need more time. On RNA, we have a very similar structure. We have a phosphate, so PO4 3 minus, in this case 2 minus. And there's a double bond on one of these O's as well. And it's connected to carbon 5, which is connected to carbon 4, carbon 3, carbon 2, carbon 1. So far it's exactly the same as the DNA subunit, and it's connected to a base. The difference for the bases for RNA is that it can be uracil or adenine, cytosine, guanine. And for DNA, it's going to be thymine, and then the other three as well. Now, as far as structure goes, this object has a very similar set of extra atoms around it. All of them are the same except for at carbon 2. Carbon 2, RNA, ribonucleic acid, has a hydroxyl group. And so that's the difference between DNA and RNA. This has the hydroxyl group, it has it, so its name is just ribonucleic acid. DNA is missing, and so that's why it gets deoxy in its name. It's missing that oxygen that should be right there, all right? If you look at these two, we can separate them out into their respective pieces. Here's your phosphate, here's your 5-carbon sugar, and here's your base. Phosphate, 5-carbon sugar, and base. And if I wanted to draw the entire structure, I would draw this. I could also draw the structure of the base. But instead, what I tend to do, and what you see in many other places, is a simplified version of this. Phosphate right there, circle representing, or sometimes a pentagon representing the sugar, and just the word base, or one of those 4 letters, T, A, C, and G, or U, A, C, and G. And this is a really simple way to draw that. And this object is called a nucleotide. This is also called a nucleotide, and it would be the same. Phosphate, base, sugar, and one of them is a DNA nucleotide right there, and the other one is an RNA nucleotide. And so we get, when we look at these objects, we get an idea of their simplified form, but things can get more complicated than this. DNA is, and RNA is typically a polymer. And a polymer is a molecule or an object that has many subunits combining together to form one thing. Just to show you, I know you guys have this in your textbook, but this would be the full detailed structure of a nucleotide. And this is the simplified structure of a nucleotide. And again, you guys have these handouts. You have them either on D2L or you have your course notes, so you can take a look at these in a little bit more detail. All right, so now that we have this, we have our simple nucleotide structure, we're gonna get a little bit more complicated. So we're gonna take these and we are gonna start to combine them together. So, if I have one nucleotide, I'm gonna draw my nucleotide this way. I'm gonna make it slightly more complicated by drawing the full 5-carbon sugar. And I'm just gonna put, as its base, I'm just gonna put the word base for now. We're gonna stick with that, okay? But I'm gonna draw the hydroxyl group. And so this is my simplified nucleotide. I'm gonna work with this and we're gonna start building a chain, a polymer of nucleotides. And my hydroxyl group is important. The reason I drew that and I missed everything else, missing everything else is just because I'm lazy. The hydroxyl group I drew because it is important. This is the site of the reaction for when the next nucleotide comes in to join in place to form what's called a dinucleotide, so 2 nucleotides. So a phosphate from nucleotide number 2 is going to come in. And actually I'm going to draw nucleotide number 2 in a different color. So there's your phosphate, carbon 5, carbon 4, carbon 3, carbon 2, carbon 1. There's my oxygen. There's my base. 1, 2, 3, 4, 5. And the phosphate, remember, has some oxygens coming out of the side. It's not just phosphorus, it's PO4, possibly with some hydrogens as well. This molecule is going to attack that bond right there. So I have this in a handout. This is absolutely in your textbook. It's for sure in some of the videos that I posted online, and it's right here. So this phosphate for nucleotide number 2 comes in. We get an attack. We get one atom attacking the bond, and we have removed from this reaction a water molecule, so very similar to when we link two amino acids together. And we have a phosphate group in between two ribose sugars. This is called a phosphodiester bond. And the reason it's called a phosphodiester bond, you have phosphorus in the middle, and you have phosphorus connected to an oxygen connected to a carbon, that's an ester. An oxygen connected to a carbon, that's an ester. And so you have a diester, so phosphorus diester, phosphodiester bond. And after I do this, I can come down here, I have an OH, still over there. I can bring a third nucleotide in, and I can build a trinucleotide. So I'm going to take this molecule right here, and I'm going to build a bigger version of our nucleotide chain, but I'm going to do it in the simplified way, just because, you know, for the sake of time, you guys have already been watching for 16 minutes, so I'm going to try and make it fast. So I'm going to do this. There's my phosphate. There's my sugar, phosphate, sugar, phosphate, sugar. I'll put 6 of them. I think 6 is a nice healthy number for what we're going to do next. 1, 2, 3, 4, 5, and 6. And here, attached to my sugar, I'm going to put one of the 4 bases. So we'll start with adenine over there. We'll put cytosine next. Why did I pick adenine? Why not? First letter of the alphabet. It's the first one that popped into my head. Cytosine, same thing. We'll just do that, and then I'll just continue, and I'll throw some other things in. So we'll do that. Cytosine again. We'll do a thymine, a guanine, and a thymine. There we go. Why did I pick this order? Why not? Any base can come before or after any other. There's no limitation on what order the bases can be, other than the organism that you're working with and the piece of DNA that you're working with to make the new piece of DNA. And we're going to see that in the next section. There is a code that matters, but there's no rule to say that you can't get a certain sequence. You'll see this exact sequence here, even though it just came up off the top of my head, you'll see this in your DNA code. If you were to do a full analysis, you would see it thousands, hundreds of thousands of times. We could actually calculate how many times we expect this sequence to pop up in all of your bases, or in all of your DNA code. Now, that's one nucleotide chain. DNA is not a single molecule. Typically, it can be, but we often get it bound to another molecule. And it's bound, if you watch the movie, hopefully you remember, it's bound according to the code. So on the other side, I'm going to use the same marker. On the other side of A, we would always have, unless there's a mistake, a thymine. There would always be a thymine over there. Next to cytosine, there would always be guanine. So here, cytosine, you can guess what's going to come next. Next to thymine, you can guess, or you should know what's going to come next. We have it right up there, the same pair, A, next to guanine, and then next to thymine. And I'm going to draw this, I'm going to draw it relatively quickly. This is the way that I would do it if I was in class, just to kind of make it quick for you guys. There are my ribose sugars. There are my phosphates. Okay, so I have 1, 2, 3, 4, 5, 6. I'm drawing 6 nucleotides, and I have 1, 2, 3, 4, 5 phosphates. Now, if we were in class, what I would be doing on the board is I would be putting my phosphate right there. When I put my phosphate right there, I'd back away from the board, look, ask, how does it look? So imagine that I put my phosphate right there. How does this molecule look to you? If you didn't watch the movie, you might not know. If you didn't watch any of the videos, you didn't do any of the readings, you might not know. But that should be ringing some alarm bells. You should be thinking to yourself, that's not right. Or if I tell you it's not right, you might be able to go and find why it's not right. If you watched the movie, hopefully you did, you might remember Crick saying one molecule goes up, the other molecule goes down. They're running in opposite directions, and he used pencils to show. So I'm showing it like this. These molecules should be running in opposite directions. So if I have a phosphate at the top of this chain here, I should have the phosphate at the bottom of the other chain. Because what we have, what I simplified here, what I did to make my job easier, is I made your job harder. You have to visualize the 5-carbon sugar here, and they look the same, and they're not the same. This one has this structure, carbon, 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 2, 1 oxygen with a phosphate right there. That would be that shape with all of the hydrogens and the hydrox or whatever else you need. This piece here, this ribose sugar here, is instead going to be the opposite direction. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, phosphate, oxygen. Notice that this and this are flipped. They run in opposite directions, so you cannot treat this as the same thing if you put the phosphate on the top or the bottom. It is not. It is very, very different. All right? So now that we have this, we also have connections in between the two chains. So between A and T, there's going to be some connections between them. There's going to be what we call hydrogen bonds. Between C and G, there are going to be hydrogen bonds. And we're going to have different numbers of hydrogen bonds in between them. And you have a handout, you have it in your course notes, and I have a really good version here. Between cytosine and guanine, we have 1, 2, 3 hydrogen bonds. Between thymine and adenine, we have 1, 2 hydrogen bonds. So the connection between these two is stronger than the connection between these two, and that does play some roles in how we study DNA, how we work with DNA. If you're doing this in university, you'll often need to memorize sequences that are A and T rich, that have a lot of A's and T's, because those are usually where the DNA is going to pull apart more easily, and it's often the starting point of some of the reactions that we do or that we study. So there we go. So I'm going to fill that in on my diagram. I have A and T, 2 hydrogen bonds, C and G, 3, C and G, 3, A and T, 2, C and G, 3, A and T, 2. So here I have a 6 nucleotide stretch and a 6 nucleotide stretch. And so this produces my strand of DNA. Each of these pairs, each of these groupings is called a base pair. So this AT here is called a base pair. Every one of these is called a base pair. And when I see this structure here, I see it drawn, I can very easily, very readily understand what the directions are just because I know it, because I've worked with it. But this is really hard. If it's 1960s or 1970s and I'm first working with DNA and I'm trying to get information to some friends on the other side of the world and we don't really have fax machines that work yet, he can't afford one or she can't afford one, how would I get this information to somebody just by typing it out? How would I communicate to them this structure of DNA? I can't easily send all of this, I can't easily draw this picture and mail it to them. That's just going to take too much time. If the molecule is way bigger than 6, I might be talking about a piece of DNA that's 1,000 nucleotides long, and that's not really easy to do. So if we look at this, the way that I would simplify it, and if we were doing this in class, it would take a bit more time, but we're just going to make it a little bit faster. So if I wanted to simplify this, I would have to keep in mind some pieces of information. I'd have to have the code. So I'm going to read this code first. Here's one chain, ACC, TGT, and I'm just going to write it this way because this is typically the way we would type on a typewriter. So ACC, TGT, and then to make it easy for me, I'm going to put its matching chain right underneath it. And the person that I'm working with knows, is going to understand what that means because I'm going to tell them, I'm going to give them the tools they need to translate this. And so I have that. Now that's fine, but if somebody else comes and looks at this, they may not know. If they didn't get the same instructions from me that my friend got, they might not know. They might not know if the phosphate was at the A side here. Maybe the phosphate was here. And maybe I drew my picture upside down. Maybe I had it drawn a different way. Maybe I didn't even draw a picture. Maybe I just wrote the sequence this way, and I didn't tell them which side was which, because this end of the molecule is a different end than that end. It behaves differently. It's chemically different. What's important about this molecule going forward is that this end has a hydroxyl, this end has a hydroxyl, This end has a phosphate and that end has a phosphate, and those are different. They behave differently. So what I'm going to do is I'm going to take this. I'm going to take this structure and I'm going to use that to inform how I tell somebody the direction. I'm going to use those numbers that I just gave you. Carbon 5, 4, 3, 2, 1. Carbon 1, 2, 3, 4, 5. And those numbers are here. That's carbon 5. That's carbon 4, that's carbon 3. It's not, I drew a circle, but we know that that's what that circle represents. So this one is the same, 5, 4, 3, and then all the way down, 5, 4, and that's carbon 3. And so I look at this chain and I look at its carbons, and the A is at the end where carbon 5 is, and the T over here at the bottom is at the end where carbon 3 is. And so I'm going to identify that right here at the A just before. I'm going to put a 5 and then the prime symbol. And that just means that it's the 5th carbon of that sugar. And here at this end, I'm going to put a 3 and a prime. And I'm going to do the same thing for my other chain. So here, the T of my other chain is carbon 3. That's carbon 4, that's carbon 5, 3, 4, 5, 3, 4, 5. And I'm using what we call the backbone. So there's the backbone of DNA. This is an unbroken chain of phosphates and carbons. Phosphates and carbons, phosphates and carbons. And I can use that as my identifier. And so here at the T side, I have a 3 carbon. So right there, I'm going to put the 3 prime. And here at the A, I have a 5 carbon. And so I'm going to put 5 prime. And so I'm going to use this to identify my molecule. Now, when I want to write about my molecule, I could write this. 5 prime A, C, C, T, G, T. That matches that. That's right. I could also do this. T, G, T, C, C, A, 5 prime. That's right as well. There's nothing wrong with that. These two are the same. Even though one is written this way and the other one is written the opposite direction, it's still the same molecule because the numbers tell me where I'm working. Am I working top to bottom? Am I working left to right? That doesn't matter. It matters the sequence and which carbon is which at each end of this sequence. So another way that I could write this molecule is A, C, A, G, G, T with the 5 over there. I'm writing this one backwards with a 5. I didn't even talk about the top strand. It doesn't matter. Why would I pick that one as the most important? It might not be the most important. So any one of these are fine as long as you put the direction identifier. I don't have to put 5. I can take that 5 out and I could put the 3 there. And that would still be correct. There's the 3. 3, TGG, ACA, 3, TGG, ACA, and we're done. When I talk about these molecules, I talk about them in what we call directionality. So here I have the 5 prime end over here and the 3 prime end over here. And so if I move this way down this strand, I'm said to be moving 5 prime to 3 prime. If I move in the same top to bottom, but I move down this strand, I start at the 3 prime and I end at the 5 prime. And so if I move down this other chain, don't even worry about that one. I move down this chain, I'm moving from 3 prime to 5 prime. So that's the direction. And moving in one direction down a double helix, you have to pick which strand you want to talk about because you're moving 5 to 3 and 3 to 5, one way for one and one way for the other. So we have to specify. And you'll see this is not just an exercise in your patience and numbers. When we do the next week, we're going to start making copies of DNA, and that direction really, really becomes important. Okay? So a couple other things before we're done. I'm going to call it very soon. This diagram is in your course notes. It is a wonderful one. It shows the oxygens, the phosphodiester bonds. It shows the number of hydrogen bonds. Take a look at that. And here's another one that you have that gives a whole load of information as well. So make sure you take a look at these, make sure you read them, come back to my instruction if you need. If you have any questions, let me know. And I kept it at half an hour. That's far longer than I thought. It's a complicated topic. This would take, 3-4 days in class from start to finish. So I hope that you're able to get through it and it makes sense. Thank you very much, guys.